with the collaboration of the Promotion Agency for Gran Canaria Island Council, within the activities of the REBECA-CCT project funded by Save Seaweed Coalition and the Interreg-MAC REBECA-CCT project. The aim of this seminar is the presentation of some initiatives that are being carried out in Europe on the cultivation of macroalgae in the open sea, its processing and the development of biomass applications. In addition, we shall present the objectives and results of the REBECA-CCT project and the role that macroalgae cultivation collections play in the current scenario of global change. For those who do not know me, I'm Antera Martel, co-scientific director and curator of the Spanish Algae Bank collection. I will be moderating this session together with my colleague Juan Luis Gomez Pinchetti, scientific director of the Bank of Algae. Without further ado, we shall start this seminar, which takes place a few days after the presentation by the European Commission of the important initiative entitled Towards a Strong and Sustainable EU-Algae Sector, which aims to maximize the potential of algae, increase production, consumption, algae products and their sustainable use in Europe. The document also addresses the role of algae as a blue carbon sink and the development of biofuel standards generated from algae, which can contribute to achieving EU targets in terms of decarbonization. In this promising context, it is a pleasure to introduce our first speaker. Dr. Fernandez holds a PhD in biological oceanography. She has worked at the Norwegian Polar Institute and at the Geomar Heimels Center for Ocean Research in Kiel. She is currently leading her own research group at the Alfred Wegener Institute. Her expertise includes phytoplankton ecology and primary productivity, marine carbon dioxide removal, artificial and natural upwelling systems, the impact of climate change on ecosystems, carbon and nutrient biochemistry, carbon export, and microbial functional diversity. She has participated in several high-sea research expeditions from the Arctic to the tropics. She is a member of the Ocean Artificial Upwelling Project and co-founder of Seafields, as well as its lead scientific advisor. Dr. Fernandez is going to talk about aquaforming sargassum in the open ocean to produce seafood, raw materials, and carbon dioxide removal. Thank you very much, Antera. I'm going to share my screen. I'm going to speak to you in English because this is what I normally do in my talks. If you then have any questions in Spanish, I can also answer them in that language.
So thank you everyone for being here. I don't need to introduce myself because Antera did it already very nicely. And today I would like to talk about aquafarming sargassum, and you will hear a lot about this algae in a minute. So first, I want to remind all of you that right now, the pathway that we are heading to in terms of CO2 emissions, if we want to get to net zero by 2050 or even by 2100, we not only need to dramatically reduce our emissions and avoid new emissions, but we also need to remove the CO2 from the atmosphere. So even the most aggressive decarbonization pathways depend on significant levels of carbon removal and storage. Between 5 and 15 gigatons per year, we need to be actively sequestering. And once we're done with that, we need to start removing the historical emissions, which is around 1,500 gigatons of CO2 that is in the atmosphere, to go back to safe levels of CO2 in the atmosphere. Not only that, we also need to decarbonize our chemical industry. So just as an example, right now, plastic production and disposal worldwide emits 1 gigaton of CO2 per year. And in order to achieve what has been pledged by the Paris Agreement and others to get to net zero, we have to decouple the CO2 emissions from the production. So instead of using fossil fuels for producing plastics, we need to start using renewable feedstocks like wood, like other plants, but also like seaweed. So seaweed can help to both of those. It can help with carbon dioxide removal, and it can help to substitute products that right now are being made by fossil fuels. Now, the areas where we can do seaweed aqua farming are usually on the coast. Most of the seaweed species available are benthic seaweed species. And so in this map, you can see in red the native range for wild seaweeds, and in green the countries that are already farming seaweed, and in a range of blues the different nitrate to phosphate ratios, which would indicate the areas that would be good for seaweed aquaculture. So it's quite a lot of space. It's 48 million square kilometers of coast. However, the coast is also used for many other things. We need it for recreation, for fishing, we have marine protected areas, etc. So it wouldn't be feasible to fill all these areas with seaweed. So if we want to really upscale seaweed aquaculture, we need to move offshore. There is no other way around it. Now, how does it look like in terms of nutrients available in the open ocean? So as you can see here on the left, you have the surface chlorophyll A, and you can very clearly see that there are areas of the ocean that are very productive and other areas that are known as the subtropical gyres that are not very productive. And this is mainly due to the nutrients available there. And you can see on the right the surface nitrate concentrations. And you can see, for example, in the Atlantic, that most of the North tropical and subtropical Atlantic are depleted in nitrate at the surface. So if we want to make those areas useful for seaweed aquaculture, we need to bring nutrients from the deeper layers to the surface. So one idea that is out there is artificial upwelling, which basically would bring nutrient-rich deep waters to the sunlit surface to enhance productivity. There are different ways of doing this. There are wave pumps driven by solar or wind power. There are also other types that are recently being developed using the density gradient of the ocean, and I will talk about that later. However, all of them, what they do is they bring this water to the surface and make the nutrients available for phytoplankton or macroalgae. So this would be useful to restore the ocean's productivity and also enhance fisheries. And only if we can sequester the carbon that is also being brought up with the nutrients with a higher carbon to nutrient ratio than what was coming up, only then we can have active net carbon sequestration. And that's important to remember. So we chose to work with sargassum, and sargassum is a very special seaweed. In particular, I'm talking about Sargassum fluitans and Sargassum natans. And those two species, they are free-floating their entire life cycle. They reproduce by fragmentation, so they do not require any complex hatchery process. As you see, they float because of the bubbles that they have here. And only when you burst those aerosis, then they sink very rapidly to the deep ocean. And also what I think is very important, besides that they grow very fast, is that they have a very high carbon to nutrient ratio. Therefore, they're very efficient at carbon sequestration. And also many algae are very tasty and they maintain entire ecosystems, but sargassum seems to be rather a habitat builder rather than a food source. So where does sargassum come from? Sargassum originally comes from the Sargasso Sea. However, in 2011, due to changes in the North Atlantic Oscillation caused by climate change, Sargasso escaped the Sargasso Sea, came to the Canary Islands actually, and then went south again with the Canary Current and established itself across the entire tropical Atlantic in what we know now as the Great Atlantic Sargassum Belt. And these huge masses of mats of sargassum form every year. They have a seasonality and they form every year from West Africa to the Caribbean. Unfortunately, of course, because of the currents, many of those of that biomass ends on the beaches of the Caribbean, causing lots of problems with the coastal ecosystems and also with the society that lives from tourism. Now, in the Sargasso Sea, there's a lot of studies, and actually the Sargasso Sea is a protected area. Sargassum is creating what they call a golden forest, and so it's rather a habitat and not a food source. So there are a few studies showing that very little invertebrates and fishes actually feed on sargassum, and if they feed on it, it really makes a very small percentage of their diet. So how does sargassum's carbon cycle currently look like? When it reaches the Caribbean coast, it kills seagrasses, coral reefs, mangroves. So all the CO2 that those coastal ecosystems were sequestering is not sequestered anymore because they're dead. And then when it lands on the beach, it starts decomposing and it starts emitting CO2 and also methane. So if we can avoid that sargassum from beaching, we're actually avoiding quite a substantial amount of greenhouse gas emissions. And if we can use it to create products and store the carbon in products or use it as fuel, then we're of course displacing products that would otherwise be emitting CO2. The problem is in the Caribbean that there are a few companies that have started working on using sargassum for products. I have here two examples from the company Carbon Wave that works in Mexico. They have, for example, an emulsifier to make creams. So right now, all emulsifiers are made from fossil fuels. This is one of the few ones made from seaweed. And they also have, for example, vegan leather that they're starting to create from this. However, the problem is that because they have such a patchy influx of their raw material, the sargassum, they cannot really upscale. And so that's why farming sargassum might be a good idea. And I'll talk about this in a minute. So there's a new company that was formed a year ago that I'm a co-founder of called Seafields that has the aim of aqua farming sargassum. So basically domesticating this floating seaweed for different purposes. So sargassum, as I said, is great at capturing CO2. And the idea would be to sequester that carbon that is fixed in the biomass, either in products together with Carbon Wave, for example, or by sinking the biomass to the deep sea. And in doing that, we are, of course, taking care that while we harvest the sargassum, we don't damage any other species. And we're, of course, monitoring and tracking how much CO2 is sequestered, how much gets to the deep sea, and how much will be remineralized down there. Now, the big vision is to create big, very big open ocean aquafarms because we want to get to scale. And so just a rough back of the envelope calculation, if we want to sequester 1 gigaton of CO2 per year, which again is not that much considering we have to sequester between 5 and 15 gigatons per year to get to net zero, we would need to create aquafarms that cover an area of 90,000 square kilometers, which is roughly the size of Portugal. So it seems like a big area, but if you see the huge areas that the Sargasso Sea and the Great Atlantic Sargassum Belt cover, it's actually not that big. However, of course, this would be more a densely packed area with sargassum, while the sargassum that is wild in the Great Atlantic Sargassum Belt is pretty much spread. So it's very difficult to harvest it because it's so far apart from each other. Now, one of the projects we just got funded through the German SPRING-D Innovation Agency is, it's called CCOS, and it's to put the carbon that is sequestered from the sargassum in those aquafarms, to process it, to transform it into ethanol. And in this case, the ethanol would be used by the chemical industry to create long-term plastics. For example, the plastics you use in your computers, in your phones, in your cars. So I'm not talking about packaging or things like that, but rather plastic that would last at least 10, 15 years, and then it would have 1 or 2 recycling steps. So the carbon would be stored there for some decades. Now, in terms of how far we are with the aquafarming infrastructure, we did this year the first trials in St. Vincent. So you need to keep in mind that we're talking about free-floating aquafarming infrastructure, which is non-trivial to get working. So these are the first images of the first prototype. So this is relatively small. This is 10 meters on an area of 79 square meters. But one of the problems we had was that when the wind and the current were not aligning, the aquafarm would collapse. So we're now trying to figure out in the next steps how to make it stay open all the time. We're also learning a lot about how to monitor the sargassum, and for that we're mostly using drones from the surface combined with divers or snorkelists in the water. So as you can see here, the thickness of the sargassum mats can be very variable. It can be between 7 and 80 centimeters, the depth of the sargassum. and the weight per square meter is between 1.7 and 14 kilos. So it really depends on how the wind goes. It will accumulate in one area of the farm or not. And so this is important for us to understand when we try to quantify what our standing stock is. We're trying to monitor it with drones using multispectral cameras so that we can identify the depth and also how healthy the sargassum is. Now, in terms of the artificial upwelling, we need that the water that reaches the surface is warmed up, because if it's not warmed up, it will sink again, and therefore it will not be useful for our floating sargassum. So that's why we're developing a stomal pipe. So the stomal principle is basically based on using the density, the salinity temperature gradient in the ocean. So you have at the surface warm, salty water, and at depth, cold, not so salty water, rich in nutrients. And so when this cold water goes up and this warm water goes down, the warm water, so they go in different tubes, the warm water basically is warming up the cold water, enabling to rise on its own. So you don't need external energy to make this pump work. It's a little bit slower than other types of pumps, but it doesn't require external energy inputs. So we're right now in the modeling steps together with Geomar, and we will try to then do a first prototype by next year. Small prototype, of course. Then the idea for long-term carbon sequestration in the deep sea is to process the sargassum, extract the nutrients so that the biomass that is left is mostly carbon, and that we compact it and bale it like you do usually on land with agricultural waste, and then basically drop it to the deep sea to around 4,000 meters depth. So we assume that because we're removing the nutrients and we are baling it, the decomposition rates will be very, very low, but we haven't measured them yet. And we know, for example, from the Sargasso Sea that in the Sargasso Sea, there's 4 times more sargassum in the floor than at the surface, which means even when it sinks naturally, it doesn't immediately decompose. So it doesn't seem to be easily decomposed. And you can see here one of our trials where we had the first bale sinking on its own, and we measured the speed. And I mean, this was only a 5-meter trial, but doing the calculation, it would take such a bale 4 hours to reach 4,000 meters depth in the middle of the ocean. We are, of course, planning the first environmental impact assessment, because we know that this can have, especially when we move to scale, can have impacts in the deep sea. And of course, we want to prevent any negative impacts. However, I always like to remember that we need to compare those potentially negative impacts to the impacts of doing nothing and continuing with climate change as it's happening right now. Now, in order to be able as a company to sell carbon removal units or carbon credits, there's a whole process that needs to be a few steps that need to be done. And so the first is, of course, to select your carbon dioxide removal strategy, which I just described here. Then you have to identify and monitor the removed carbon fate, do a life cycle assessment. So we have to take into account also how many emissions we emit while doing the whole processing. And for the examples I just showed you, we're about 9 to 10% of the carbon we sequester, we're emitting again from our emissions, which is really good compared to other methodologies. And then we have to do constant monitoring and verification and reporting, and then you can have your carbon removal unit that you can then sell. So we're still far away from that step, but that's the goal. Thank you very much for your attention, and if there are any questions, I'm happy to answer.
There are no questions in the Q&A right now, but I would like to ask you a question. I know that there is much debate on the issue concerning seaweeds which are produced to be sunk to the bottom of the sea to remove carbon, and that these seaweeds are not used in the industry to generate products for food or for biomaterials, etc. What is your view on these issues, which are often found on social networks? Basically, it is a problem of scale and what type of seaweeds we're talking about. There are seaweeds like kelp, for instance, that, among other things, are very good for nutrition. Sargassum is not good for human consumption, as I said, and neither is it good for animals. So there are types of seaweeds that are good for specific things. I think the solution will be for that part of what harvest from the aqua farm will go to products, and another part will go to carbon sequestration. If you have a process like ours, where valuable elements are extracted, like nutrients in our case, we can sell them as fertilizer, or we can put them back into the marine environment so that those nutrients are not lost, and what is left can be sunk. In the end, the decision will be determined by where the needs are and what the market says. If the most important thing right now is to replace all plastics, then we can focus on ethanol production. And if the most important aspect is to sequester carbon, that it is sequestered for at least 1,000 years, then the sinking part will be given priority. I think that we can do everything, that we don't have to choose one or the other, as these options can go hand in hand in the same way as can be seen for coastal seaweed farms, like kelp, for example. These calculate that 20% usually sinks by itself and sequester carbon, so why not do the same in the open ocean? I just think that you can do both. That's my point of view. Fine. Gabriel wants to thank you for your presentation and asks if it will be available. We are recording this session and we will let you know when it will be available on the Banco Algae platform.
Fanny pregunta how much funding that you need to support this progress.
So there are two different avenues of funding. One is, of course, the basic research that needs to be done, and that is usually funded by public funding. And I mean, we have right now, I would say, around $1 million per year. We would need way more to move faster. And then if you start talking about how much do we need to have the first prototype up and running, we're talking around 20 million. So that's what we're looking for the next funding rounds. Yeah.
Right. And another person is asking, have you developed any standardized protocol for estimating the quantity of carbon sequestered through sinking of seaweeds?
So we are currently developing it. We have let's say a written protocol on all the steps that should include, like, for example, monitoring the air-ocean PCO2 exchange, monitoring the weight and the amount of carbon in the biomass itself, and then monitoring the respiration rates in the deep sea or the remineralization rates. So we know all the steps that we have to take into account. We have tested the ones at the surface already and know that work. We haven't tested the ones at the deep sea yet. And we're applying for a methodology with Vera, for example, which is one of the verifying agencies, to get this standardized. That is not public yet.
Okay. And
I think that Juan Luis has a question. Mar, is the model you're using for sargassum relevant for other seaweed species? You always compare it with laminaria, which are not brown seaweeds? I'm not sure. I don't know about other types of floating seaweeds, but if it's a seaweed that needs a substrate, then the answer is no. In our aquafarms, we don't have any other types of support. It looks like a ranch than a farm. The idea is to keep the sargassum in a closed space. We are not providing any kind of substrate or anything similar. Maybe for some similar seaweeds, the answer to your question could be yes, but for all the others, it's quite different. Thank you very much, and congratulations for your presentation. Thank you.
So the question was if this model of farming would be useful for other types of algae, and I was saying that maybe yes, but only free-floating algae, so maybe ulva in other circumstances, but not for kelp or any other benthic macroalgae. So that's what I was saying. And then the other question that was in Spanish was, I don't remember.
No, I think it's. It was my questions about the use of cultivating algae and then sinking without using the biomass.
Yeah, and then there's a question I see about impacts.
Yeah.
Should I answer that one? The one from Ines?
Just a short answer
Very
in
short.
English.
Yeah, I will answer in English so that everyone understands it. So the impacts of sinking sargassum, the only big impact that we can think of is, of course, if oxygen would be consumed when it gets remineralized. But as I said, we're doing everything we can so that those remineralization rates are as small as possible, and the carbon actually gets sequestered for millennia. So we're trying to minimize that, but that would be one thing. Another impact could be that it actually creates like structures where deep sea life could grow on. Like when a ship sinks, they also are full of things growing on it. So it might even be beneficial in terms of enhancing or giving a substrate for deep sea organisms. And then about the upwelling pipe. So the impacts are basically that you're bringing nutrients to the surface, so you can you will get primary productivity. You will get probably phytoplankton and also seaweed growing. So yeah, that's how you change the surface basically. So you convert a system that was oligotrophic into a more like nutrient-rich system.
Gracias, Mar. Pregunta Inés, ¿qué impactos esperarías que tenga hundir el sargassum en el fondo marino o utilizar la bomba para generar el upwelling? Ok, si es la misma, aprovechando la pregunta anterior. How can we ensure that the carbon capture through the seaweed is stored for more than 100 years underneath the sea? How can we monitor carbon degradation rate in the seaweeds?
So when you sink the seaweed to 4,000 meters, it is sequestered for more than 100 years by definition, because even if all of it would be remineralized, those waters would not reach the surface again for at least 1,000 years. And that's assuming that everything would get remineralized, which is not what we expect. So that is for sure longer than 100 years. And then how can we monitor the remineralization rate? That's basically measuring oxygen consumption. And so that's what we will try to measure in the deep sea using landers and sensors. And the concentration of nitrogen and phosphorus in the upwelled seawater, we're talking about 35 micromolar nitrate, and I don't remember the phosphate out of my head, but it's in a Redfield ratio, I think.
Muchas gracias, Mar.
Thank you very much, Mar. Perfect presentation and answers for the purpose of this talk. Thank you very much for being here, and please stay with us, if you can, till the end of the seminar. Of course, I will. Thank you very much. We now turn to Dr. Rui Pereira. Dr. Pereira is a biologist. He's currently head of the unit of marine algae at A4F, Algae for Future, a biotechnology company located in Portugal with an experience of over 20 years in the field of algae production at an industrial scale. Prior to this position, he has worked as research director at Alga Plus, a company which he co-founded in 2012 and where he has held different positions from project manager to production manager, quality assurance, among others. Moreover, Dr. Rui is a member of the Portuguese Committee for the Standardization of Algae and the products derived thereof, as well as member and co-chair of the expert working group in macroalgae farming within the ECLIPSE project, the European initiative to bridge politics and biodiversity-related knowledge. Dr. Rui is now going to talk about the EU ECLIPSE report, Macroalgae Cultivation and Ecosystem Services. Good morning, Rui. Good morning, Antera.
Thank you, first of all, for the invitation to be here and the chance to present this work from ECLIPSE. So I'll be talking on behalf of the expert group that made this report. ECLIPSE, not exactly representing A4F, but it's a pleasure to be here. For those of you that don't know ECLIPSE is it started in 2016 as a project, a EU project, and then because of the success that it had, it moved into a sort of an autonomous entity, ECLIPSE. The main objective is to answer questions that can be posed by different organisms, policy, governments, the EU, of course, but also NGOs or companies. Like I said, to answer questions related to biodiversity and ecosystem services. So the work group concerning macroalgae cultivation and providing climate-related and other ecosystem services was a group that was created based on a request by the DG Maritime Affairs of the European Commission. So they basically approached ECLIPSE and said, we would like to know more about how can we use macroalgae cultivation to control climate change and to see if all the news and the research and everything that we see nowadays in different aspects of our social life, social networks, etc., if that has a scientific support. So once they receive a request for somebody, and if the request is selected, the ECLIPSE management body puts together a call for experts, so people can apply. And if you are selected, we will then start working during a few months and try to answer the request. That's it. The good thing about the organization is that the ECLIPSE has a very active, a very supportive management body that explain to us, the experts, that we don't know how these things work, explain us how the process goes. But it also has a very good method expert group, so people that are not necessarily experts in seaweed or in other kinds of organisms, but they are experts in different methodologies that they explain to us during the first meetings, and then we make a selection of how do we want to approach the question and try to answer it. So basically, in our case, we decided to use two different methods to try to answer the question by the requester, the Delphi methods and a quick scoping review. Antera, your hand is raised. Do you want me to stop for some reason?
No, I think it was
okay.
raised previously.
Before.
Yeah, I will take it off.
So the Delphi method is basically a method based on questionnaires that are sent to selected people. And the quick scoping review is just one of the possible ways of reviewing the scientific literature. Both methods have pros and cons, and you can see there. For the Delphi method, and both methods in this case, they run parallel. Okay, we execute them at the same time. The Delphi method, basically, we identified more than 100 possible respondents from different sectors of the society, and we set up a small questionnaire based on, of course, based on the question that we needed to answer, and we sent this questionnaire to these experts from different sectors. Like I said, we try, at least in the invitation group, the invitation group was very well balanced, based on people from academic and research, people from the industry, from NGOs, from professional associations like aquaculture, the AVA, different professional associations, but also from international organizations of other kinds. One of the cons of this method is that it relies a lot on the availability and on the responsiveness of the experts. Okay, so that was the problem that we faced in this case. The method usually starts with one general questionnaire, sometimes with very open questions even, where the responders, the expert, will list their opinion. Those that first round of questions is then grouped and organized, and there's usually a second or eventually a third round asked to the same people that will then try to rank the questions from the first response. This is just to give you an idea of the composition of the group in terms of respondents. Unfortunately, during the first round, out of more than 100, more exactly 120 something invitations sent, we only had 22 responses. And unfortunately, even more unfortunately, in the second round, we actually had only 6 answers from those 22 initial respondents. Nevertheless, we did try to organize the questions, and based on how much they were referenced and how they were ranked in the second, we also tried to rank in terms of importance given by the experts. Okay, so in this table you will find the different ecosystem surfaces that were mentioned by the experts, and now they weight the different ecosystem services in terms of importance according to the experts again. In blue, you'll see the ecosystem services are related with provisioning. In orange, the ones that are related with the regulation. Okay, and in green, the ones that are more related with cultural and aesthetic ecosystem services. This is according to classification from the EPBS. You see that what comes up immediately in terms of ecosystem services: macroalgae grown as food, as regulation of water quality, as feed, and then different kinds of ecosystem services. In terms of potential negatives of trade-off from macroalgae cultivation in large scale, these are the points that are also related with ecosystem services, and in some cases they were pointed by the expert. You can see them there. The first of all being conflict with other uses, at land or at sea. We don't, in this case, we did not discriminate if the macroalgae cultivation would be in near shore, offshore, or inland. Okay, and then the second one is unknown environmental impacts in deep sea, but not just, but also in pelagic, in pelagic systems. Okay, another, the third and the fourth had to do with the concerns about genetic changes, but also on the mismatch in supply and demand of biomass. A little bit of what Mario was telling before, that it's still not clear in the market what is the main purpose for which the large-scale cultivation should be carried out. That is also because the market, at least in the Western world, was still developing. Very important also in terms of knowledge gaps identified by the responders. The Delphi methodology had to do with knowledge gaps still on farming technologies. You see there, and within farming technologies, or in these kind of technologies, subcategories identified without weight, in this case of importance: strain development, consistent of production quality, and developed of mechanization. But then also a very important group of knowledge gaps has to do with market data and economic data to support the large-scale cultivation of macroalgae. And then you see other points that are mentioned with not so relevant to the responders. But keep in mind that most of the responders, if you remember the pie chart that I showed you in the beginning, most of the responders were from academic and research, and not so much from the other sectors that we also try to engage. And of course, this naturally bias the responses, okay? This is what we had, and those are the results that we could show and share with you. In terms of the quick scope review methodology, this was based on a research of keywords, okay, in those databases, Corpus and Web of Science. The search was done in the middle of 2021, June 2021, based on those keywords, or actually 6 combinations of keywords, as you can see in there. And then there were naturally exclusion and inclusion criteria for the different papers that we found that went through actually 3 phases of selection. And those were the phase 1 inclusion and exclusion criteria. These are phase 2 exclusion and inclusion criteria, first based on the title and abstract, and later on in the third phase based on full text. It's a very thorough method in which each paper was actually reviewed by at least two experts within the Eclipse group. And in case 2 experts did not agree in inclusion or exclusion, a third expert will then review the same paper and issue a final decision. So the result was like this. So we started with 1,200 papers selected, and then the number was being decreased, filtered, let's say, through phase 1 to phase 3, and we ended up with 280 papers that were then thorough review, and from which we extracted information, as you can see in there. So year, location, species, the type of farm, the size of farm, the ecosystem services, the constraints, if they were identified, and the negative impact or risks, if they were identified. The data that I will show now, it does not necessarily help to respond to the question posed, but it was interesting data that is also a nice picture of how the research and the work is being carried out in the world in terms of distribution through genus or species, but also geographically. So you can see there that out of the 280 articles that were eligible, we see 37 genus and 77 species, okay? But in Europe, because the project was also, or the answer was also to be focused as much as possible in Europe region, we find only 11 genus comprising 17 species that were being subject of research focused on this kind of matter. Currently, there are actually 4 non-native species that are being thoroughly studied in European waters. Now, based on the geographical distribution of the research and the species, it's interesting to see how this can be a nice picture of how the efforts in terms of distribution through different genus is being made throughout Europe. So, as you can see in Asia, see a very well-balanced distribution of research between different genus of species, and those are the most common ones in terms of aquaculture nowadays, maybe except for the ulvalis, but it's not so common in terms of volume of production aquaculture. Latin America also very even distribution, but then if you move to Africa and Oceania, you see most of the papers, the research papers, are based on the chromatoids, so the ones read out that are basically used for extraction of carrageenan, a source of phycocolloids. And if you move to Europe and North America, not surprisingly, a lot of the research is based on brown algae, the kelp. In terms of this is the average of the complete, so without the breakage in terms of geographic distribution. So very interesting, and this starts already pointing to some of the difficulties in terms of conclusions and actually some of the knowledge gaps, is that when we try to divide the scale of the research or the scale of the data from which the data was obtained, we see that most of the research is being based on land-based or near shore, but within these different locations, very little is being done and published at a scale that we consider relevant. And this is the scale that was at least over 1,000 square meters, and even this is not a very large scale if you want to actually have a meaningful conclusion for impacts that, like Mar was explaining before, for something that needs to be carried out in the scale of thousands of square meters or thousands of hectares of area of production. So this is clearly a limitation of knowledge, scientific knowledge that exists nowadays, that most of the published papers are based on either small or medium scale research. In terms of ecosystem services that were proven by the the papers, the 280 papers. See that most of them have to do, again, with provisioning and regulation. Not so much attention is being given to the cultural ecosystem services. Within the provisioning, you see there that most of the attention is being paid to seaweeds as food, feed, and hydrocolloids. But in terms of regulating, most of the research is being focusing on water quality and then biological regulation, and not so much on climate regulation aspects. Okay, so it's clearly another gap that we can find in those papers that we consider. In terms of constraints, there was a match between you can say it's a match between Delphi and the quick scope review in terms of the importance of the technical and the methodological constraints that exist. But you can see that in the research literature, we can see that the environmental constraints are actually much higher than what we see in Delphi, in the Delphi method. In terms of negative impacts that we could identify from the papers, you see that there are quite a few listed, but there's a very large, more than 3/4 of the chart pie, it's for papers that did not investigate any negative impact. So clearly, the research that is being carried out these days, until middle of 2021, is not paying a lot of attention to negative impacts. So it's not that they don't exist. It's clear that the research and the projects are not focusing on potential negative impacts. And the ones that we have in there listed, a lot of them, like the algal blooms, are exclusively coming from a series of papers that is being carried out in China, or in Chinese waters, or in that region, because of the famous green tides that are known to occur there since the last decade or so. So that the weight of those algal blooms as negative impacts is very much based on that series of literature that is being produced in Asia. So as a sum up of these, we try to organize the different this different information in a diagram that could be easily interpreted by policymakers, because at the end, as you remember, all this is to inform the European Commission and DG Mare on how to proceed in the next steps of programming, policies, organization, and decisions. We also try to organize the data in what we call a pestle analysis, so try to group the different information in terms of political, economical, social, environmental, and technical aspects. And we can see there some information or some summary of what are the main ecosystem services that are identified, the main potential these services. But again, there's a big gap in terms of what are actual negative impacts that could come from very large scale macroalgae cultivation or seaweed aquaculture. And then we have different weight in terms of technological constraints, different weight of environmental, social, and economic. As you can see there, the numbers actually represent the number of papers that refer to these constraints. Okay, and if you sum up those numbers, it can actually go higher, but some papers refer more than one constraint. You can imagine it's also possible. Now, keep in mind that this is the results of the quick scoping review and the combination of that, but also with the methodology, with the Delphi method. Both methods have some constraints, pros and cons that I showed in the beginning. Naturally, the bias of based this only or exclusively in research papers is that all the information or decisions on what to do and what not to do was made by the researchers, okay, with the interest of the researcher or the projects that were supporting that. And so some of those technological, some of those gaps, constraints, they not necessarily match the gaps and the constraints referred by SMEs that are involved in the seaweed aquaculture. For instance, it's very common for those of you that go to a conference and talk with other SMEs and people having to deal with regulations concerning the commercialization or the large-scale cultivation of seaweeds for commercial purposes. One of the things that always come up in this discussion are the legal aspects, political aspects related with regulations. Okay? Naturally, because the Delphi method was mostly answered by researchers and academics, because the research papers are produced by the researchers and academicals, political and regulational aspects are not necessarily the main focus of this work. So we can see that the legal aspects have a very small share of this pie. But like I said, when we talk with people in different conferences and different events, we see that's still a problem. There is references by a lot of the SMEs and the people in the industry. To finalize, as main conclusions, in terms of knowledge gaps and some open questions that we suggested to the request of the DGMR and the European Commission, things to have to do with site selection. So which environmental parameters define a suitable site for implementation of a sustainable seaweed farm. And these, as we've seen in the previous, have to do a lot with nutrient availability, but also with a lot of other factors that have to be accounted for when we select a site for seaweed cultivation. The scale of cultivation, okay, how does the scale of seaweed cultivation affect the ecosystem services provided? And we know that if the scale is too short, the impact in terms of ecosystem services will not be meaningful. So we have to really talk about the very large scale of cultivation, and we don't have enough information about a lot of the challenges and the impacts, positive and negative impacts of large-scale cultivation. So this is something that needs to come up in the next few years. But large-scale cultivation is very unlikely to come from solely research projects. Okay, large-scale cultivation probability is that will come up from the initiative of companies, such as the one that Mar mentioned before, and others, because they will be the ones that will eventually be able to gather enough funds or invest private funds for the large-scale cultivation. So it will be important to have a match between those projects that could arise in the future, hopefully, in large cultivation, with proper research and academic experts that will follow up or that will go along with these projects and, for instance, actually be able to monitor the actual impacts, positive and negative impacts of such projects. And then there are technology knowledge gaps. It will be important to answer some of those technology knowledge gaps. Otherwise, the large-scale cultivation will probably never happen. And of course, there are also some economic and environmental questions that arise, and they also be important to account for when we plan those large-scale cultivations. If we don't have in mind those questions, it's very likely we'll not be able to. We can have the large-scale cultivation projects implemented, but we'll not be able to answer the questions that we'll still have these days. So this basically was more than a year of work. We have much more data, but this is how I can summarize, let's say, this information in the time available for this. And of course, I'm available for questions now, or if you want to reach me by email, my contacts are in there. Feel free to do so at my time. Thank you for your attention.
Muchas gracias, Rui.
Thank you very much, Rui. The picture you've presented of macroalgae cultivation and utilization worldwide is very interesting. You have partly answered my question about what you would expect if you could have answers instead of from academics, mostly from people who are involved in medium to large-scale cultivation in different parts of the world, and especially how local policy may affect the development of the industry.
sorry, in English. The knowledge that I have from that, it's also biased by my own knowledge. We, as much as possible, try not to give that personal information in the project, even though in the actual report at the end there's a section that is clearly our opinion as experts. But to answer your question, my experience, and I've been in the research world for many years, and in the last 10, 12 years, I moved into the SME, and more industrial. And I've been talking with a lot of people trying to do the same. And clearly, if more people from the industry would reply, I would certainly see more weight in terms of the political and legal aspects that we haven't seen, because that is always a question that arises in terms of licenses for production, license for commercialization, regulation in terms of quality standards. There's a standardization process ongoing in the EU that will eventually hopefully will help solve some of those issues. Hopefully will help solve them and not making them more complicated, but that's a discussion for another topic. And if we go along also to different areas of the globe, in some areas there are ecosystem service related with social aspects are much valued than we do in Europe, for instance. SMEs from Asia, from South America, and research from those areas point a lot to ecosystem service related with social, cultural, and aspects related with the empowerment of women, better quality of life for different sectors of the society. And in Europe, we tend not to give so much importance to that kind of ecosystem service, more related with cultural or aesthetic purposes. So the only thing that was agreed, and for, of course, if you think about more answers from NGOs, I'm sure you would have much more weight given to the environmental aspects and to the potential negative impacts that macroalgae cultivation could have in terms of ecosystem services, so-called these services. So from the research point of view, there are still some questions raised about that, most question marks and potential, but not so much was being actually carried out, focusing on those potential negative impacts. And that did not show up as much also in the Delphi methods. So a more balanced group of answers from these sectors will probably actually make the much more balanced information about the gaps, because now they are focused essentially on technological and economic, and not so much on the political or the environmental, as we see. That will be the main difference. But again, it was not our role to say to DG MAR and the requester, okay, you should focus first on technical, then on political, then on regulation. That is not the purpose. Our purpose was just to present the information as we see it available now, what is certain, what is potential, what is not so available, and it's up to the policymakers and politicians to actually make the decision what is the actual priority. If, as we know, it's not easy to focus on everything at the same time. If they are not able to focus on everything, they will make their priorities, their decisions.
I think that Juan Luis has a question.
Very interesting talk, and well, a lot of results coming from this report. But I would like to ask you, related to this last communication we have from the EU regarding the importance of the development of algae or cultivation in Europe. What is your opinion related to this communication, this last one?
I have to admit I haven't read it very carefully. It still didn't have time to incorporate some of these data, but it's very clear to me that the EU it's really committed to the development of the industry. I'm sure that will happen. I would like to see more connection. Hopefully, we'll be able to do that in the next framework program more connection between the industry, and we keep talking about this, but it's still true, more connection between the industry and the companies. Because it's like we saw before, Maris is carrying out a project and looking for a solution to be able to grow the sargasso in very large areas. When we talk about answering problems like climate change, we are talking about very large areas. And we see that we don't have any knowledge on how to manage and what are the actual impacts of these large areas. And a very large area of cultivation will not arise from a research project. Okay? That's very clear. A research institution will not have the capability, and we're not talking just about funds, okay? We're talking about logistics, knowledge, practical knowledge, engineering, and all that. A research project will never be able to carry out a very large-scale cultivation just for the sake of identifying the potential positive or negative impact. All the research will be done to a scale that is manageable and possible and financeable. So this large-scale project will have to arise from private initiatives. So the link between the private initiative and the research to be able to follow up and to monitor and to quantify the result and apply the scientific knowledge to the actual practical knowledge of production, cost of production, and selling. Coupling these two things parallel will be, I think, very important to be able to gather more information, and hopefully in a few years it will be much more clear if seaweed can actually be an answer to climate change. I like to think that they could. My personal opinion at this point is that we are much more closer to be able to use seaweed, as Mar was explaining before, as a substitute for fossil fuels, and that will already be a very important achievement, I think, for the next few years. As she also explained, that's not enough, but if we could replace a lot of those fossil fuel, and the seaweed could be raw material for plastics and for many other applications. If we could do that in the next 10 or 15 years, then I think we are very close to do it, because the technology exceeds I mean, we are able to do a lot of different products, and most of the time it's just really a matter of availability of biomass at a cost of production that makes that suitable for plastic. Because everybody sees projects that are able to produce, like Mario was saying, leather, plastics, many different aspects, but then all these different technological developments, they go into the barrier of, okay, but producing this kind of plastic and this kind of leather is still much more expensive than producing the plastic from the fossil fuels. So at the end of the day, it's an economic decision. And so we need to scale up, at least to be able to have more of these raw materials and to be able to use seaweeds to offset or to substitute some of these fossil fuels. I think that's already a very important step towards the control of climate change. And then the next step will be to explore ideas such as the one that Mar presented before. Why not? And as long as those things, a lot of people, and I'm sure she is aware of that, a lot of people criticize that idea of sinking the seaweed. If the seaweed is valuable for other things, I think we should try to use them for other things first. But her approach makes a lot of sense. Like in terrestrial crops, there are things that are not useful for a lot of things. Why not use them for that purpose and make the proper research and investigate? And why not? Why should we discard that from the start? I mean, I think it's important, but as she will probably agree or not, we're still the way to apply what she's doing for climate change it's a longer way than what I referred before, that is produce plastics, produce green fuel, produce leather, produce not even talk about food and feed and all other things that could be obtained in a much more sustainable way from seaweed. But go back to your question, I think the EU and the document makes an effort in that direction. Let's see if we keep the effort going. Some people criticize that we're going through a hype phase about algae and seaweed, and a lot of people are afraid that it's just a hype up and down.
Yeah.
And what I see from these documents from EU is a good sign that it's something that's here to stay for the next few years. And if we're able to do it right, I think it will really stay. It not be just a fashionable thing like would happen with the microalgae and with fuels 10 years or 15 years ago. I hope it's here to stay. And the document is certainly a good sign.
Yeah.
A good sign of that. It's a sign also for the private investor. Hopefully it will have the positive impact that we expect.
Very good. Thank you. Okay, Mark.
Sí.
Yeah, just briefly to mention that, of course, it will make a big difference if the cultivation happens in the coast, because then it's easier to use the material for products. But if it starts happening in the open ocean, then transporting the wet biomass or even the dry biomass, if you have to dry it at sea, that's very costly. And that's why in those scenarios, sinking starts making sense economically speaking. But I fully agree that from the development, it's still way further down the line than making products.
Thank you very much. Rui, I would also like to ask you from a personal point of view, or from the point of view of the Spanish Bank of Algae, do the technical problems involved include the study of biodiversity, the preservation of biodiversity, the availability of the material concerned?
Yes, some of those questions arise because it's very clear from at least from the quick scoping review that the industry is still based on 10, 12 species from 5 or 6 genera and not more than 12. Some people say up to 20 different species. That's very short for a sector that needs to develop and needs to adapt to climate change that is also happening we also feel that in the sea, in the ocean, and needs to adapt to different geographical locations. And if we want to avoid, and I think we are already doing that, introduction of species from one place to the other, it's very important that we are able to gather more knowledge on the actually algae diversity or the biodiversity more related with the algae or the seaweeds in different regions, and start to explore more of that. I mean, we all know there are more than 10,000 or something species described worldwide, and we are based an industry on only 10 or 12. So we really need to diversify. We need to study more about, look for other species that could be applicable to the aquaculture. Sectors like pharmaceutical, cosmetics, the chemical area, they have a lot of knowledge about seaweeds, algae, and different compounds from those that can be extracted from those seaweeds. But they base that only on natural populations. And they have all the information sitting in their desks, but they know a lot about what can they extract from that species and that species. So if that information reaches the industry or researchers like yourself, it will be possible then to explore more seaweeds that they already know that they are valuable. Because right now we're exploring just a very small fraction of the diversity of seaweeds that exist. So if we diversify, we certainly need more projects like the seaweed bank or the algae bank that you manage, because it will be important to have strength. It's what happens in agriculture, right? I mean, we have seed banks, we have projects storing seaweeds for many years. We have companies specialized in producing seeds. We have to evolve in the same way. I think there's no other way to try to learn from the good practices of terrestrial agriculture. Terrestrial agriculture has a few thousand years of advance in terms of seaweed aquaculture, but we have to learn from at least the best practice that they have and the way they organize themselves, and the way they specialize themselves. And we have to go in the same direction. Hopefully, we should go much faster and we will not need 2,000 years to be at the level that agriculture is nowadays. And those kinds of projects and information, it will be strain development, optimization, and we're not even talking about genetic modifying organisms or nothing like that, because we don't need to go there, at least yet. We have so much to explore. And in fact, what the market tells us, at least the food and the feed market, is that the consumers don't want that coming from seaweeds. They actually run away from genetic modified organisms. But we do need some optimization, some strain optimization. We do need to know more about different strains in different geographical locations to be able to progress much faster. Yes, and initiatives and entities like yours will have a very important role, I think. And they have to come up in different regions of the world, hopefully.
We fully agree with you. Thank you. We try to coordinate all these projects with companies, and it's sometimes difficult because these companies that invest in research and development are reluctant to make their data available, even though it's clearly desirable. Despite this, we contribute through projects such as those that Juan Luis is going to present now, and we thank you very much, Rui, for presenting this report, and we'll kindly ask you, just like Ma, to stay with us throughout the seminars. We shall welcome your feedback. I will introduce my colleague, Dr. Juan Luis Gomez-Pinchetti. He's the co-scientific director of the Spanish Banco of Algae and permanent lecturer at the University of Las Palmas. He has an experience of over 30 years in research, member of the Spanish committee, like Rui for the Portuguese one, for the standardization of the algae sector and the products derived thereof. His research focuses on physiology, on the cultivation and biotechnological aspects related to algae, seen from an applied point of view. Today he's going to talk about the Algarden, Innovalga and Seaweed projects as promoters of seaweed cultivation actions in the Canary Islands. Thank you very much, Antera, and thank you all for joining the webinar. I'm going to talk a little bit about what we at the Spanish Bank of Algae have been working on for the last 30 years, related to the production of macroalgae and microalgae in an environment such as the Canary Islands and from an applied point of view. The summary of this talk will be a review of these types of actions that we have been carrying out over the years and what we are currently doing in relation to these projects, focusing on the production of macroalgae, mainly because we're working on an issue related to carbon sequestration. We are also carrying out actions in this regard. For those of you who don't know us, we are located on the Tagliardi Docks on the island of Gran Canaria. In these infrastructures that you can see on the slide, we are developing different ideas and projects ranging from the conservation of diversity, as we spoke about a few minutes ago, to the characterization of species of both macro and microalgae from an applied point of view, from a biotechnological point of view. In an environment like the Canary Islands, where we are surrounded by the sea on all sides and have sunlight 365 days a year, we can develop these types of projects and activities always from the point of view of innovation and knowledge transfer for the development of the sector in our immediate environment. All these years we have been working on the development of intensive cultivation systems for macroalgae, mainly native to the Canary Islands, bearing in mind that there is a high level of biodiversity in the Canary Islands and that the populations are small in size, so they're not susceptible to being exploited from an industrial point of view. Given the large number of species available on coasts, we are particularly interested in the development of production systems for red and green algae, since from the point of view of intensive cultivation, brown algae are much more complicated for various physiological reasons. So we have focused on the development of these intensive production systems for green and red algae, which are the ones we can see in the example. The development of intensive cultivation systems, mainly in cultivation tanks, allows us to work on the selection of species and the systems control, as Rui mentioned earlier, evaluating the possibilities offered by the control of the parameters that influence the production system in terms of biomass quality. Not only as far as productivity is concerned, but also related to metabolism synthesis under different environmental conditions within the various cultivation systems. We're always looking for the sustainability of production systems, that they be sustainable over time. We work a lot on vegetative cultivation, and we also work on controlling the quality of the biomass we produce depending on the different types of applications. In recent years, we've also felt the need to control traceability because we're talking about time-consuming processes, and therefore we need to know the traceability of the production systems and the systems we work with. I should mention the work we have done over a long period of time in the development of integrated multi-trophic aquaculture systems, where we have worked with the use of wastewater from production systems for organisms of higher trophic levels, mainly fish, and the possibility of using macroalgae with a system of microfiltration of wastewater, which has allowed us to characterize all the parameters related to assimilation and biofiltration processes from different types of wastewater, both on a pilot plant scale, working with photobioreactor culture tanks, and also at the level of raceways production. All this has given us the possibility of acquiring important knowledge concerning different species of macroalgae, not only from a physiological and biochemical point of view, but also from the point of view of production, with the possibility of obtaining production that is sustainable over time, over several years, evaluating the quality of the biomass, and above all, we're very much interested in the possibilities of obtaining biomasses with specific features and which are interesting from an applied point of view and of great interest for the industrial sector. Within the examples, on a small scale of different applications, we have characterized all these species from different points of view, and always looking for industrial applications, for example, as a source of food, for organisms that are also produced here in the Canary Islands, such as aliotis. And recently there has also been a lot of interest in the use of algae in gastronomy, with questions concerning the use of algae in food preparations. We have some very representatives and high-level examples, such as Chef Abraham Ortega, who last week was awarded a Michelin star and is a connoisseur of the possibilities of using algae in our environment. In recent years, we've also worked within the BEA-ULPGC platform to assess the development of the algae production sector in the Canary Islands, which has allowed us to identify bottlenecks related mainly to regulatory issues, such as the regulations that affect the development of the algae production sector in the Canary Islands, and it has allowed us to assess the possibilities for the development of the sector in the Canary Islands. By extension, we're also developing projects related to these possibilities in the Macaronesian region, carrying out projects such as REBECA-CCT with research labs in Madeira, the Açores, Cape Verde and Mauritania, and always with an eye to the possibilities that are being developed in the European sector due to the interest in the development of the micro and macroalgae sector at European level. In recent years, from a specific perspective, considering global change, as we have already seen in the talk given by Ma and some comments made by Ru on the subject, in the latest projects that we are developing, we value this type of action. We are not only focusing on the development of algae production and algae cultivation systems, but also on how we can make the techniques more sustainable, and always working with the objective of reducing, within our possibilities, the effects that our activities can have on climate change. In this sense, we cannot forget all the information we receive regarding the possibilities that algae offer us in this revolution. Because this research does not only concern new sources of food, but also the ecosystem services they provide, especially in terms of the possibilities they offer for cleaning wastewater, but in particular all the processes related to carbon sequestration and the possibilities of reducing the effects of climate change through the use of algae. In this regard, in the last 2 years, we have been working on a series of projects mainly based on the production of seaweeds. And one of the main projects on Brazilians, supported by the Spanish government, is the InnovAlga project, which is a project related to the promotion of an emerging and sustainable sector for macroalgae in Spain, and which is mainly based on the diversification of species and the development of applications with high commercial and environmental value. And we cannot forget these two aspects: the possibility of working on ecosystem services while producing a biomass that can be of industrial interest on the market. Within InnovAlga, we are working with different types of species that are new to us. In some cases, we had done some previous studies, but in the InnovAlga project, we are starting with the production of this type of species, as we can see in the examples. In the first year of the project, we are focusing on Caulerpa and Valonia species, which are well adapted to intensive production systems, to cultivation tanks, and develop specific morphologies and growth characteristics. And we are currently analyzing not only these characteristics, but also the value of the biomass from these 2 points of view. What possibilities do we have from the point of view of product development and from a commercial standpoint? In the coming months, we will be working with other species that we had evaluated some time ago and that could be of great interest in this type of application, such as Aliopitis incurva or Ipnea spinella, red algae species that are of particular interest to us due to their biochemical characteristics and their potential for different industrial applications. And within InnovAlga, I would like to stress the importance of focusing on any development concerning environmental sustainable solutions. One of the examples we are most proud of in recent years is the possibility of using recycled CO2 in all our facilities through a gas cleaning process based on gas produced industrially in some industrial areas of Gran Canaria, and which is recycled, cleaned, and with food grade that we are reusing in our facilities in the cultivation systems. We are making use of small ideas and small possibilities, considering sustainability and adjusting to the possibilities in terms of sustainable development techniques in this field, in the algae production sector. Another project that I would like to highlight is a cost action called Seaweed, which values all the knowledge that we have developed in Europe and in many countries around us related to the production of ulva in different cultivation systems. We bring to the project mainly our experience in the development of culture or tank systems based on all the results and experience we have acquired in the development of filtration systems for wastewater derived from fish farms and wastewater from ulva cultivation. The overall objective of seaweed as an innovative aquaculture model derived from overproduction is based on the knowledge we have of its biology, the development of production systems, the industrial applications related to food, animal consumption, and other types of polysaccharides products, pigments, etc., that can be obtained from this biomass, the possibility of working with bioactives from the ulva biomass produced in this type of cultivation system, the valuation of ecosystem services, and finally, taking into account all the social, regulatory, and political aspects for the development of macroalgae culture in Europe. With the experience we have gained in these previous projects and that we're currently developing, we are also starting a project called Algarden in collaboration with the Port Authority of Las Palmas de Gran Canaria and the university to develop a prototype algae cultivation system in the open sea to help us mitigate and quantify the possibility of using algae cultivation systems to sequester carbon related to port activities in such an important environment as the port of Las Palmas. To this end, we have already talked previously about the importance of the sources that generate carbon dioxide or greenhouse gases that affect the development of climate change. And one of the actions and one of the most important sources is port activity, mainly at European level, which contributes to the carbon footprint and affects climate change. Based on the data available, we have reviewed and re-examined the principles that help the development of open sea cultivation systems, which are large cultivation farms, such as the one we see in the slides, a macroalgae cultivation farm in China, which has also been developing multi-trophic aquaculture systems, a production farm that produces not only algae but also other types of organisms within the concept of integrated multi-trophic aquaculture. When we were preparing this project, we also revisited the initial ideas developed in the 70s, both in the United States and in countries such as Japan, with projects such as the Wilcox Project and the Sunrise Project, which were based on the development of large ocean farms for the production of biomass for energy production. These projects at the time were pioneers in today's large ocean farms that can generate and help mitigate the effects of climate change. And based on this idea, we have moved on to new developments related to the use of algae as a carbon sink in large ocean farms. And as Ma mentioned earlier, the possibility of using this type of carbon sink to move the biomass produced to the bottom of the ocean or to remove the carbon to the ocean floor. Well, having said this, developing this type of approaches to for the removal and filtration and removal of CO2 from the atmosphere, when we look at the numbers that are published, the results are really encouraging. In this table, for example, we can see that the algae produced in Asian countries in terms of potential CO2 sequestration in tons of dry weight of biomass per year, or the carbon sequestration processes in this type of approach. And with all these numbers, and given this scenario, we have developed an idea using ropes for the cultivation of algae species, such as ulva, of which we have considerable knowledge. We know the macrobiological aspects involved, we know how we can fix and prepare the cultivation ropes to install them in this prototype pilot system that takes us out of our environment, the work on land in cultivation tanks, and allows us to work in cultivation systems in the open sea. Within the environment of the port of Las Palmas de Gran Canaria, we will assess all the carbon incorporation processes related to the growth of algae on the physical cultivation surface and on the cultivation ropes that we are going to install on this pilot platform that we have already prepared on the Las Palmas dock. The idea is to assess the productivity and growth of the algae in the system, both in the cultivation belts and in the cultivation surface area that we have available, and make calculations based on the valuation of the carbon sequestration from the biomass produced. In this project, there is no possibility of sinking the biomass to the ocean floor because we are talking about an area which is relatively close to the coast in a port environment. However, we are evaluating the possibility of using all the biomass we produce in different types of applications. On the slide, we have included some of the most attractive ones, the possibility of using the production of biofertilizers for applications in agriculture, or as biomass for obtaining fuels or other metabolites of interest for industries such as cosmetics, biomedicine, and obtaining polysaccharide pigments. Bioplastics seem to be very interesting for us, and as we develop the project, we will be working on developing this subject as well. This is an interesting project that opens new doors because until now, this type of research had not been carried out in the Canary Islands. It is a new activity that can give us a lot of information about the real possibilities for the development of this type of system along the coasts of the Canary Islands. I would like to end my presentation here, and I'm available to answer any questions you may have. Thank you very much, Juan Luis. I think that just as Rui mentioned about the need for a relationship between companies and what they are discovering, the needs they're finding and the possibility that algae can provide a solution. This is where these projects can provide an answer. And are there also companies participating in the Innovalga project? The Innovalga project involves technology centers, and the members of the consortium are CT Aqua, which is the project leader, and FACO, the University of Cádiz, and we as the University of Las Palmas de Gran Canaria. But there are companies interested in the developments and results that we are going to obtain from the processes that we are generating. Companies mainly related to the food market, as well as agricultural biofertilizers, and the issue of obtaining metabolites for certain types of biomedical applications. And what role does legislation play in this regard for human and animal food and for biofertilizers? At the moment, and this is one of the questions I had earlier for Rui, I think that the role of regulation is currently very important in the development of the sector. In the Canary Islands, we thought that it could be a significant limiting factor because there is no traditional use and there are no industrial activities related to the production of seaweed. However, it is now an emerging, a new sector, and lately many European initiatives such as EU for Algae, for example, and other initiatives are assessing what is the current situation. As far as the regulation of the sector is concerned, the idea is to transmit to the European Union the need to establish rules and regulations that allow for the development of the sector with concrete actions. In the last few meetings we have dealt with this issue frequently, and there is a lot of interest in standardizing the regulation of the sector. What I was saying is that what I'm seeing when talking to companies is that there is this type of rhetoric. How many tons are you going to provide me with? What are the bureaucratic and legislative problems I'm likely to face? And what kind of algae are you offering that are not traditionally used? This is a major stumbling block for companies. You're right. We have the example I mentioned of the BEA-ULPGC Gran Canaria platform, where we had made an important effort to attract activities within the sector on the island of Gran Canaria and by extension the Canary Islands. And despite the interesting initiatives that promoted the development of the activity on the islands, we came up against a major limitation, which is the regulatory issue. For the application to obtain permits, the administrative procedures are complex and take a long time, precisely because there is no experience. The regulation is closely related to the development of agriculture, mainly animal farming, and so when you talk about algae, you ask for a permit to develop business activities within the production of algae, and the administrative procedures remain complex. From my point of view, and we are dealing with this in the framework of Innovalga, the aspect of the homogeneity of algae and regulation, we are realizing that in many regions of Europe, the same problems arise. That is to say that there is no clear regulation that allows the sector to develop smoothly, and we believe that this is an aspect that we will have to deal with seriously in the coming years. And as far as the novel food, novel feed regulation is concerned, the novel feed regulation provides a standard norm in Europe that everyone has to comply with, and although it is a complex regulation, it is quite clear, and we know that there are some examples related to both macro and microalgae production that are achieving this novel food certification. One example being Tetraselmis chui, which has been incorporated into the novel food catalog in Europe. And this regulation is clear despite its complexity from a technical and economic point of view. In order to apply for a permit and to be included in this regulation, it is economically complex. Still, the regulation is clear. In the future, there will probably be a simplification of the novel feed regulation related to algae, because there are many projects and there are many possibilities associated with algae.
Solo para, just to add, related to that, Santera, the problem with the novel food, it's based on the regulation that Juan Luis was mentioning, that refers that we need to prove that they were being used before 1997 or something like that. And that's particular critical for the microalgae, because it's difficult to prove that they were being used before that time in other areas. And then up to recently, we thought that this could be a problem also for the seaweeds, because in Europe, we don't have the tradition to use seaweeds in many places, aside from islands like the Canary Islands and the Açores in Portugal and the other places. So everybody was concerned about that. Why do we have to follow that regulation? Because we don't have an historical documents proving that, while in other places, there's more than 300 species being consumed for centuries. And the interpretation that I know now that it's given by the EU is that apparently the historical proof then don't have to come from European Union, from the European region. So if we can prove that seaweed, and this was an answer in one conference a couple of months ago that was posed to one of the officers from the EU, exactly because of that. They said, well, we don't exclude historical proofs of the seaweed being used in Japan. So if we can prove that it was being used in Japan, they are open to accept it also in here. And that alone opens the door for many other species of at least for seaweed, that could be easily introduced. The fact is that some of those species that were not part of the European list of approved species, they were already here because they were being imported from Asian countries. So at least for seaweeds, I think the problem will, for now, that will not be a strict problem, but it will be in the future, like we were doing before. We have thousands of species, and we have only a few hundred that we have historical records in Japan or in China or in Korea that have been used. If we want to use more and exploit more of the diversity, we need to expedite somehow this kind of process. And one thing is for food, another thing is for other applications, which should be, and it's part of being within European Union. All the regulations, it's important they are made, they are created with the objective of promoting or supporting the industry, but we must not forget that at least in the beginning, the effect will be the opposite. At least the immediate effect will be to slow down while people are adjusting to the regulations or not knowing what are the regulations. So hopefully in the future, the regulations, once they are in place, they will actually have that effect of promoting the industry. But in the beginning, they tend to be more an obstacle than a facilitator. The other day I was talking with a colleague from South Korea. We were discussing exactly about kind of regulations we have in place here, and he was saying, you know, if we had those regulations in place in Korea 40 or 50 years ago, the seaweed industry would not be what it is today. Now, I don't want to sound irresponsible. I don't mean that this would be like the Far West and everybody or the jungle. But the truth is that there's this trade-off between being in the front of concerns and regulations regarding food safety, regarding climate, and EU, it's usually in the forefront of these things. The trade-off is that all these industries that are new, they tend to have more obstacles to the implementation. Hopefully in the future will change. And I mean, in In Korea, they are now putting in place regulations regarding debris and waste from seaweed aquaculture. Of course, all those things are important, and you should have them in place. They should have them in place before. But the truth is that they didn't have them, and the industry developed very quickly, and now they are trying to put in place regulations. But those regulations are being created with the notion that an industry already exists, and they cannot afford to destroy that industry. So they are being in place in a realistic way, in a responsible way, and people will have to comply. Of course, they will have a few years to comply with those things, but they are being created and from zero adapted to that kind of activity. Not like when we were saying, we are doing sometimes in some countries in Europe, adapting the animal aquaculture regulations to seaweed aquaculture, or adapting other kinds of regulations to seaweed aquaculture. That's the tendency of that. I don't think it's the better way to do it.
Completely agree with you.
But that's where we are now. Hopefully, in the future will be better.
I've heard of some companies interested in developing some specific cultivations that are not contemplated as novel food, novel feed, or a new biofertilizer that also has its own legislation, who would like to join together to meet the costs of the whole process, which is long and costly, of including this new algae and Cianobacteria or macroalgae in the novel food list. This is also what the platform we belong to, BEA-ULPGC, is trying to do. I would like to continue asking. I just wanted to ask a small question. How is the standardization committee that you, both Rui for Portugal and Juan Luis for Spain, are part of in Europe, contributing to this trend? The work is progressing. There are some standards published in Europe related to activities linked to the Standardization Committee for Algae and algae-derived products. There is still a lot of work to be done. There are different working groups related to different fields of application. And the group of experts working within the committee is convinced of the need to develop this type of standard in order to have an industry that has weight and that the consumer can trust. The development of these standards will be fundamental for the development of the sector in Europe.
Yeah, absolutely agree. I just add that when we're talking about standardization, we're talking about a process that is volunteer. So we're talking about a standard process that could be adopted or not by a seaweed producer or by an algae producer. It's not the same as a regulation that becomes mandatory and everybody has to comply. And this is also important because, of course, when we talk about meeting some kind of standards, that has to have a cost and adding some complexity, some rigor to the process. But that's important. It's important because, as we know, we're trying to develop an industry in Europe that has to compete to the same industry that is being carried out for many decades in Asia. And one of the things that we know is that the prices of the seaweed and the price at which they arrive to Europe, it's much cheaper than the production cost that we have. So seaweed industry in Europe has to differentiate in some way. And one possible way would be, to produce different species and diversify the species and the offer to the market. But another way, an important way, will be through quality, through some kind of standard or a stamp of quality that could very clearly differentiate the production done with those standards from the production in other. It doesn't mean that everybody, a producer in China or in Korea, can actually adopt the standards and they can offer the seaweeds with the same standard. So the standards will not be solely for the European Union. So they want to be adopted and certified by other authorities, but they could be adopted for But what we think that it will at least level a little bit the comparison in terms of quality of the product, because nowadays it's difficult for a producer to compare or to value their product in terms of protein content. If the neighbor says it's producing seaweeds more protein, but at the end those things are not comparable. So those kind of things that seem simple, but at the same time, like Banco was saying, they are complex, and the process goes along, takes a few years, but they are important. I think they will be an important step to help the seaweed industry, to be able to differentiate and to compare and have arguments to compete with seaweed production from other sides of the world. I think it's an important process.
Yeah, I agree.
Perfect.
Okay,
Antera, I think we're slowly running out of time and that you can now make your presentation. Later, if we have time, we will also present the video. The project is Dahaleb. I would like to introduce my colleague, Antera Martel Quintana, who is the scientific director of the collection of the Spanish Bank of Algae, our curator and researcher at the University of Las Palmas de Gran Canaria in the field of applied phycology, with 30 years experience as a member also of the Institute of Oceanography and Global Change of the university, and is lead investigator for projects such as REBECA-CCT project, the REBECA-CCT, REBECA, as well as Interreg-MAC projects. She is really an expert on algae biodiversity, and most of her work has been related to Cianobacteria and microalgae. But over the last few years, she's also worked on seaweeds, and I'm really grateful to be able to share these ideas with my colleague. She's going now to talk about the results and the objectives of the REBECA-CCT project. Good afternoon. Thank you very much, Juan Luis, for your introduction. I'm going to present the REBECA-CCT project, which is actually the project for which we are holding this seminar, and which is a project funded by the Save Seaweed Coalition in its 2021 call for proposals. And given the projects we are carrying out, in which we are working increasingly with macroalgae, in fact, we are going back to our essence at the beginning of our work at the Spanish Bank of Algae, where we need biological material that is well identified, that is well maintained, and that can be used quickly and reliably. So we took advantage of the fact that the Save Seaweed Coalition has made this offer to work with algae in Europe. Thus, would just like to add that within the framework of the Save Seaweed Coalition, we have carried out research on 53 strains of seaweeds, some of which have already been incorporated into the collection, and which we are identifying from a molecular point of view. We are also developing several treatment and isolation initiatives, especially for larger seaweeds, and we have the support of the Alfred Wegener Institute, to which Mar Fernandez Mendez also belongs, and where in 2 weeks' time, staff from our laboratory will be attending a course on the isolation of reproductive cells. We would also like to thank SAMS, the Algae Culture Collection in Oban, where some colleagues will also be attending a course on seaweeds from a commercial point of view. We would like to thank the whole team of the Spanish Bank of Algae and also the Bank of Algae and Cianobacteria of the Açores, who have participated in this project and who have made this seminar possible. I thank them very much for their participation. You can always contact us by email. You can find more information on our website. And I would also like to thank our speakers who have kindly joined us to share their experience. This is the beginning of a series of webinars related to the REBECA-CCT project. This is the first webinar, and from January onwards we will start to offer you a program of webinars in which many more professionals are going to participate. Certainly as brilliant as those who have participated today. Juan Luis, do you want to add anything else? No, I would just like to thank the attendees for their participation, and Mar and Rui, who have been with us in the organization of this webinar, to all the people of the Spanish Bank of Algae, who always support our webinars. And I'm sorry, Antero, that you had to interrupt your presentation because we had much more to comment on. Yes, we could not make it today, but we will do our best to present it in the next webinar. We will leave some time for REBECA-Seaweed to present the results. Yes, because there is a lot of work behind it. We have done many things. We have some very interesting examples within the protocols that we are developing for microalgae and working also on seaweeds. It opens new doors for all these projects that we are currently carrying out, and we hope that new ones will arise. I would just like to thank you all for your participation, and I hope that we will be in touch and that we may find new opportunities to collaborate and disseminate all this information that we're generating. Thank you. In the recording of this event, we're going to include the presentation and the video we have from the University of Tunis, which we have not seen, but I think it has been a very interesting day with a very rich debate, and I think it has been worthwhile. We will thus share it with you. We will send you an email with a link so that you can access all this information. Thank you very much to all of you who joined us today. Good afternoon. To conclude this seminar on cultivation and carbon sequestration organized within the framework of the actions carried out by the REBECA-Seaweed project, I will present the project's proposal and results. REBECA-Seaweed aims to lay the foundations for the creation of a network collection of seaweeds in Macaronesia, aimed at the conservation and study of biodiversity, specifically those algae that we managed to incorporate into the catalogue to offer material which is identified, isolated and maintained alive with already curated and validated data for the development of algae cultivation and the promotion of projects related to bioremediation, algae applications and the use of biomass, always within the notion of biorefinery and circular economy in mind, within the framework of the Macaronesian bioregion, which includes the Açores, Madeira, the Canary Islands, Cape Verde and Northwest Africa, with their peculiarities from an economic point of view and from the fragmentation of the territory, which condition and make it fragile territories in a scenario of climate change. This project is financed by the Safe Seaweed Coalition and its 2021 call for proposals, and was presented by the two collections belonging to the REBECA network, a network of microalgae and cianobacteria collections in the Macaronesian region, namely the Spanish Bank of Algae of the University of Las Palmas de Gran Canaria, and the Bank of Algae and Cianobacteria of the Açores of the University of the Açores. In order for you to understand the framework and the objectives of the project, I'm going to present the proposal that was made to the Safe Seaweed Coalition and on which we have been working during 2022 through a video that I'm going to show you now.
My name is Antera Martel. I'm the curator and the scientific director of the Spanish Bank of Agriculture Collection, BEA, at the University of Las Palmas de Gran Canaria. The BEA maintains and supplies and develops programs on cultivation techniques and valorization of microalgae and cianobacteria from the scientific and biotechnological approach. The BEA and the Banco of Algae and Cianobacteria of Açores, BACA, at the University of Açores, both members of REBECA, the Blue Biotech Network of the Macaronesia, present the project REBECA-CCT. The aim of REBECA-CCT is the establishment of a reference collection of seaweed from the Macaronesian region, within both collections, which enlarges the biological capital of the REBECA network. Among the activities that will allow us to implement the project, we highlight the training of the technical and scientific staff in the isolation and maintenance of seaweed, the adaptation of space and equipment for the maintenance of the seaweed specimens, the isolation and long-term maintenance of in vivo and cryopreserved strains, the genetic identification and integration of all curated data of the strains in the metadatabase of MIRI and REBECA network, and the promotion and dissemination of REBECA-CCT among the scientific community and public administrations involved in mitigating the carbon footprint by actively valuing the cultivation of macroalgae in industrial and poor areas. We are confident that the actions will contribute to enrich the knowledge of local seaweed preservation and the offer of strains of both collections, promote the capabilities of seaweed as a raw material for the aquaculture diversification and the development of sustainable industry, and reorientate the economic activity of the archipelagos towards low-carbon models through the use and cultivation of seaweed as an anthropogenic carbon sink. We would like to thank the Safe Seaweed Coalition for this grant and its important support on the conservation of macroalgae and the sustainability of the seaweed industry.
This was the proposal that we presented to the Safe Seaweed Coalition, and these are the steps that we have taken to establish the Macaronesian seaweed collection. Up to now, as I mentioned in the video, we have several collections: the Açores collection, the Cape Verde, Madeira, and the Canary Islands collection, and we are now establishing the Mauritanian collection. We have a total of approximately 3,000 clones of microalgae and cyanobacteria strains. Through this project, we have managed to increase the species catalogue with a total of 43 seaweeds. The work has consisted in carrying out local sampling, as the work is being carried out by the Azores and Canary Islands collections, we have initially focused on the Canary Islands archipelago and have taken samples of algae that may be of particular interest for some of the projects that we are developing jointly with companies, technological centers, and research groups. The work carried out after the collection of samples consisted of the mechanical cleaning and removal of epiphytes using antibiotics and germanium dioxide for the removal of diatoms. A very laborious job, thus requiring a long time to guarantee clean clones and avoid any contamination. All this work has been carried out aiming at, first of all, the conservation of the smallest seaweeds, which are in fact macroalgae in the culture collection, as if they were other seaweeds maintained by subcultivation. Those that have meant a greater workload for us are those that are seaweeds per se. All the field data, the genetic and morphological identification data, as well as the culture maintenance data, can be found in REBECA's database. These are some of the algae specimens that we currently have in the collection: Ipnea spinella, Cauler paraquemosa, Codium, Ulva, in this case Ulva lactuca. But we are testing the maintenance of 3 local species, including in the Canary Islands biodiversity database, namely Valonia utricularis, Gracilaria cornea, etc. We are maintaining very small species, such as ectocarpels, as if they were microalgae by subculture kept in Erlenmeyer in a liquid medium. Larger algae for the initial tests and for the cleaning processes are being maintained in larger volumes in activation chambers, and for those that we are already taking to the cultivation plant, we maintain a small stock culture in tanks. This would be, for example, the case of Ulva, and Juan Luis told us earlier that we were developing the Algarden project, and these are the inoculums. Seaweeds are then cultivated in tanks and subsequently on ropes that we take to the platform where we're going to evaluate their growth and the carbon capture process through the harvesting of the biomass, and carry out an evaluation of the carbon sequestration process. These would be the ropes in the tank. Ropes were the initial tests of spore fixation and growth of the thalli have been carried out, which are then taken to the floating platform, where this pilot experience is analyzed in situ within the port facilities of the port of La Luz and Las Palmas in Gran Canaria. This is the pilot experience Juan Luis told us about, aimed at the development of a system that removes CO2 produced by port activity. And as another example of the application and supply of seaweeds to the R&D sector, including the development of aquaculture and the first experiences involving companies, we have the Innovalga project. In this project, we are working with Caulerpa racemosa, Valonia, Ibnea, Alopitis, and Codium. The aim of this project, from a more commercial point of view, is to develop trials with algae that may be of economic interest for the development of products for food and feed, and also for environmentally friendly applications through the minimization of waste that may be generated by specific industries. It is also very important to focus in this case on the recent regulations regarding the production and processing of seaweeds, both at local, regional, and national levels, as well as at European level, to establish the needs and requirements for the development of this activity. As I mentioned at the beginning of this talk, with this project we wanted to lay the foundations for the availability of seaweeds that can be used in the aquaculture industry and in research. But for this, we who come from the field of microalgae and cyanobacteria have had to take several training courses, and we have realized how difficult it is to maintain seaweeds in a living or cryopreserved collection. For these trainings, we could count on the invaluable help of the Alfred Wegener Institute through Dr. Inka Bartsch, Dr. Lori Hoffman, Andreas Wagner, and Isabel Cardoso. They gave us a training on seaweed isolation and purification through the isolation of reproductive cells. I initially told you that we started here at the Bank of Algae to carry out this process mechanically, but this involves many risks, and for those seaweeds with an apical growth, it is a valid technique, while for other seaweeds, the best method is through the isolation of the reproductive cells. A training course was very kindly offered by our colleagues from the Alfred Wegener Institute, who also promoted an important initiative with the creation of a biobank network of seaweeds through the Safe Seaweed Coalition in 2020-21. The Institute created this biobank network that gives support to its members, such as the course attended by members of both the Spanish Bank of Algae and University of the Açores. And now that we are towards the end of the training, which coincided with the implementation year of the project, we are going to visit the Marine Science Laboratory where Dr. Sophie Steinhagen works, a researcher dedicated to the cultivation and development of products derived from ulva, and who is going to provide us with considerable support for both the collection and the Algarden project. My sincere gratitude goes to all those who have contributed in one way or another so that we may reach this objective. And from a training point of view, it has been very important indeed. I would also like to thank my colleagues at the Spanish Bank of Algae and the Bank of Algae of the Açores for taking on this challenge, which has served as a starting point for carrying out very important work, especially now that Europe is ready to back algae produced in Europe. And also, given our biodiversity, we live in a bioregion that is a biodiversity hotspot. We should continue along the same line and make every effort to develop this source of wealth, considering our geographical and climatic characteristics. We must preserve it and take advantage of what biodiversity can offer to improve our quality of life. Thank you all very much for participating in this webinar, and I hope to see you in the following webinars. Given the positive response to this webinar, I hope that it will also be the case for the following talks that we will offer in the next REBECA-CCT webinars. I thus invite you to the next meeting. Looking forward to your participation. Thank you very much.